Okay, next we're going to talk about the potential addictive properties of cannabis. And of course, the active ingredient in cannabis is something called tetrahydrocannabinol. Tetrahydrocannabinol, known as THC. And if you want to be a real chemistry stickler, it's technically there's different types of THC. It's negative trans delta 9 THC. But we'll call it THC. And so what does THC do? Well, that's a good question. What does it do? Well, first let's consider, first we have to draw out here a dopaminergic neuron. All right, so here's our soma and our nucleus here. And so this, right here, we're in the, since we're talking about addiction here, we're in the ventral tegmental area here. This is where the soma of the dopaminergic neurons are, if you remember. And so I'll sort of draw in a very, well, let's, let me attempt this. We'll zoom in here. Right here, okay? This is a little synapse going on right here that will expand out here. So I'm really botching this here. Let me draw this big window, right? And so where it's kind of like this. I believe is how we might do it. Okay. And so what are we looking on this little window here on our dendrite of our VTA neuron? Here will we have a synaptic spine. And I can already tell that I'm already going to cause problems here, but we're going to go with it because you've got to learn to sort of be able to be flexible. And this is our axon terminal. And so, unlike the way that I almost always draw these, this is actually the postsynaptic neuron, right? Because it's receiving, the flow of information is this way. It's receiving information from the presynaptic neuron. Okay, and specifically in the context of our THC, which will make green, great color for THC, or the most appropriate color perhaps. And so, right, THC. Our little dots of green THC. And so this is the flow of information from right to left this time. And this is necessarily an inhibitory presynaptic neuron, because that's what we're going to see the THC acting on. Right, and again, we're talking about the addictive effects of cannabis, which are much, the risk of becoming addicted to THC is much less than those psychostimulants that we just looked at. Cocaine and amphetamines are highly addictive compared to cannabis, but cannabis can be. And so cannabis is going to respond to a cannabinoid receptor, which is a G protein coupled receptor. And so this one here specifically is CB1. And so it is a cannabinoid receptor 1. And this one here is specifically, well, here you'll see it has inhibitory effects, right? And like all the other ones, I'll keep the same color scheme. We've got our little blue G protein here that's attached, right? This is metabotropic, and so THC binds. I guess it should actually physically touch once we're exposed to THC. It binds to the CB1. And what this does here, I'm just going to draw just very general an inhibitory signal in our GABAergic neuron here. And let me write that here. Right? This is inhibitory, which means this is a GABAergic neuron. Because that's what GABA do. This is what GABA does, right? GABA is released, and it causes inhibitory signals, right? It will bind to our, and we'll make what color aren't we using? We'll make GABA red here. So GABA, right, binds to, in this case here, I'm drawing a GABA1 receptor. So a GABA1 receptor. And what that does is when it binds, it opens up and it lets chloride flow in. And that hyperpolarizes the postsynaptic neuron, making it less likely to fire an action potential. And what happens when you hyperpolarize a VTA neuron, right? Hyperpolarizing leads to a decrease in dopamine, or the decrease in the probability of dopamine being released in the nucleus accumbens or the medial prefrontal cortex in this case. And so what we do here is we inhibit, THC will inhibit the inhibitor, right? So THC will prevent this from happening as much. It will prevent as much GABA from being released. So by inhibiting the inhibitor, what it does is it increases the effective excitatory signals no longer have to compete with the inhibitory signals, which I don't know, I'll inhibitory, they're a little curly Q, inhibitory signals. All right, and so the neuron here decides what it's going to fire here in the axon hillock. By processing all these inputs here, but THC make it a little bit easier for it to make its decision, because THC will reduce the amount of inhibitory signals coming into these dopaminergic neurons, and so those neurons will fire more action potentials, releasing more dopamine. Okay. There's a GABA. There's no such thing as a GABA1 receptor. GABA A, or GABA A receptor, as opposed to GABA B receptor. GABA B receptors are metabotropic. GABA A are ionotropic, inhibitory, leading to inhibitory postsynaptic potentials. Okay, so let's talk about, wouldn't be fair to talk about addiction if we didn't talk about nicotine. So nicotine, the psychoactive chemical in tobacco. And so nicotine briefly, nicotine binds to nicotinic receptors. And what else binds to, what endogenous chemical binds to nicotinic receptors? Do you remember? Acetylcholine. Acetylcholine binds to nicotinic receptors, right? So this is what it's binding to in our neuromuscular junctions, and this is a ionotropic receptor, and specifically a sodium ionotropic receptor. So it allows sodium, and what happens if we open up channels that allow sodium, it floods into the cell and it depolarizes it, more likely to cause an action potential. Right? And so nicotine binds to this as well. And you might be asking yourself, why doesn't this cause a problem in our neuromuscular junctions when we smoke a cigarette? Because if that's the case, nicotine should be activating these receptors like crazy, and we would be having spasms, constantly contracting our muscles, and probably end up to something similar that we see in tetanus, in which we have a constant contraction leading to paralysis. And that's because there's different forms of nicotinic receptors. And so nicotinic receptors are what are known as pentamers. So their protein channels are made up of 5 distinct parts. Let's see if I can draw this relatively equally. So we want 5. There we go. And there's different forms, different types, right? So this would be one type of protein here, and another one here, and yet another one, and right, they can be repeated, and so you can have different forms in here, and there are different types. There are a whole class of alpha 1 and alpha 2 all the way to alpha 10. And then we have a beta 1 and beta 2, and I think this goes all the way out to beta 4. And then there are a few others. There's and this isn't going to necessarily be an exhaustive list, but there's epsilon, and there's actually I don't gamma, and that's kind of that's a Y instead of a gamma, but gamma. I'm not very good at my Greek alphabet here. But and delta, that's the one that I was trying to find. And so different types of subunits, right? And so this may, this might be alpha 1, and this is an alpha 2, and a beta 3, and a gamma, and a epsilon, perhaps. And so we can have different nicotinic membranes made up of these different protein subunits. But nicotine is an agonist, right? So I'll write that here. It's an agonist for nicotinic receptors. So that means it activates it for a very specific type of nicotinic receptor. And specifically, it is what we call alpha 7, that's a Z, and alpha 7 homopentamere. And so you might be able to guess what that means based on the name homopentamere. So it's all the same. So that means all of our subunits here are all alpha 7. And where do we find alpha 7 homopentamere nicotinic receptors? Well, we find them in dopaminergic neurons in the VTA. Indeed. Right here. So I'm drawing a little blib here, which is our. Let me do a better job. I'll sort of blow it up a bit here, so as not to confuse anything. So we're kind of expanding our dendrite out, a little dendritic spine here. And the nicotinic receptors are going to be here, and when activated with nicotine, is going to bring sodium inside, and we'll color code our nicotine here as red. And so when red binds to, and we'll make this different from this, our special alpha 7 homopentamere. It's our purple receptor, and when nicotine binds to it, it allows an influx of sodium in, and this, of course, depolarizes our VTA neuron, and it's more likely to send action potentials down, releasing dopamine. Okay, this will be the last example. Opiates. Of course, we'll have to talk about opiates. Among the opiates are among the most addictive substances in the world. In fact, it's heroin, I believe, is what is considered, based on a number of different factors, what is considered to be the most addictive substance. Heroin. I apologize for my handwriting. Heroin. Also, methadone is a opiate, often used as a tool to help people get off the more extreme ones like heroin, and oxycodone, which is prescribed pain relief. So we've talked about opioids already as an example of neuromodulation, but there are a number of different type of opioid receptors, but the most important one that we're interested in is the mu opioid receptor, and we've talked about this. It's the same receptor that we talked about in our explanation, and there really isn't anything too new here, but there is something right at the end that I'll talk about that makes opioids sort of a little extra insidious. And so first, the mechanism that it has directly onto the dopaminergic neurons is very similar to what we see in our cannabinoid effects, right? So I will once again be focusing in here on this box here, and so what we have here is an input onto our, and this is in our BTA, right? So this is our dopaminergic neuron. right? And this is sending signals all the way down to perhaps the nucleus accumbens or the medial prefrontal cortex. And what this does is our mu opioid receptor, G coupled. I think I need one more there. And so when this binds our opioids, and so let's say morphine, since that was the same example we used for neuromodulation, it binds here. And what it does is it reduces that GABAergic inhibitory signal, just like in the cannabinoids. So typically, and I'll draw this a little bit more accurate than I did last time, GABA is being released, and it is synapsing onto its, or not synapsing, the GABA neurotransmitters are binding to the GABA A receptors here. GABA A receptor. And we get an influx of chloride, and that hyperpolarizes. And so you're less likely to get an action potential in that postsynaptic cell, which is our dopaminergic VTA neuron. And so what the opioids do is they send a signal that prevents this from occurring. All right, and so this in turn makes our VTA a little bit more likely to be excitable, sending action potentials down. And so I mentioned that there was one other thing that makes opioids a little bit more insidious. And so what I described here, it's kind of all over the place, right? This whole process here is what we would call, in the context of opioids, the dopamine dependent pathway for addiction. So this is in the context of addiction. But there is something that's called the dopamine independent. And so this can cause addiction without dopamine. Opioids can. And so it's simply that the mu opioid receptor can bypass the VTA, right, which is what we saw here. Right, it doesn't need necessarily the VTA, though it does, and it'll indirectly increase the activation of VTA by inhibiting the inhibitors. But we also have opioid receptors, these same mu opioid receptors, in the nucleus accumbens and in the medial prefrontal cortex as well. And what they do there, the neuromodulatory effects, the neuromodulatory effects of opiates on the mu opioid receptor are the same as those effects caused by dopamine itself. So it's not, the opioids are not binding to the dopamine receptors, but it's causing the same effect. So the combination of the opiates and the mu opioid receptor is a dopamine mimic in a sense. It's causing the same neuromodulatory effects in the nucleus accumbens and the medial prefrontal cortex that you get with dopamine. So not only do you increase the amount of dopamine indirectly from the BTA, but you also double the effects because the opiates come in and they act just like dopamine in these two target areas that are associated with addiction because they increase drive and craving and seeking out the stimulus and committing a lot of resources to remembering the behaviors and the cues associated with that stimulus. So I hope you've learned a little bit here and found at least some of these things interesting in how specifically these drugs affect the addiction properties and how they hijack the reward pathway of our dopaminergic system.